Assalamu alaikum. Today we want to continue talking about the citric acid cycle. Now in this table we see energetics of conversion of pyruvate to CO2. And if you look thoroughly at this, these are the 8 reactions of the citric acid cycle and the reaction before the cycle. And most of the reactions are exergonic. The highest exergonic one is acetyl-CoA hydroxyloacetate to produce citrate, and the highest endergonic is the oxidation of malate to oxaloacetate. Notice that in metabolism there is something called coupling between reactions, which means that the energy released from exergonic reaction drives endergonic ones. Now, in addition to the energy released by the oxidation reactions, there is more energy to come from the electron transport chain. When the 4 NADH here we have, this is a summary of the conversion of pyruvate to CO2, 4 NADH and FADH2. These will be reoxidized by the electron transport chain so that a considerable amount of ATP are produced. Now, the control of the citric acid cycle will be exercised at three points, or 3 reactions: citrate to isocitrate, isocitrate to alpha-ketoglutarate, and then alpha-ketoglutarate to succinyl-CoA. So we have control prior to the cycle. So let's start with the regulatory enzymes that control the citric acid cycle. And then we have 3 within the cycle, as I said, and 1 outside the cycle. And if we look at the next figure, it shows us the control points in the conversion of first pyruvate to acetyl-CoA, and then within the citric acid cycle. Now, if we look thoroughly at these points, all of them, they are inhibited by ATP and ADH. And these are the end products of a series of reactions. And if we go back to here, also acetyl-CoA, which is the product, inhibits its own formation, and we call this product inhibition. Now, the enzyme pyruvate dehydrogenase is, however, activated by ADP, which is abundant when a cell needs energy. Now, in mammals, the actual mechanism by which the inhibition takes place is the phosphorylation of pyruvate dehydrogenase. Now, pyruvate dehydrogenase complex exists in 2 forms: the phosphorylated form, which is inactive, and the dephosphorylated form, which is the active one. Now, in mammals, a phosphate group is covalently bound to the enzyme in a reaction catalyzed by an enzyme called pyruvate dehydrogenase kinase, and when the need arises for pyruvate dehydrogenase to be activated, now the hydrolysis of a phosphate ester linkage occur, and we call this dephosphorylation. So these 2 enzymes, the pyruvate dehydrogenase kinase is stimulated, however, by ATP, and the phosphoprotein phosphatase, which is activated by calcium ions. Now, both enzymes are associated with the mammalian pyruvate dehydrogenase complex, so this will permit effective control of the reaction from pyruvate to acetyl-CoA. Now, again, the kinase and the phosphatase are found on the same polypeptide chain. Now, if we wonder here why acetyl-CoA inhibits the pyruvate dehydrogenase. Because when fats are plentiful and are being degraded for energy, their product is acetyl-CoA. So this acetyl-CoA now is plentiful, and so there is no reason to send carbohydrates to the citric acid cycle. Now pyruvate dehydrogenase is inhibited, and the acetyl-CoA for the cycle comes from other sources. Now, regulatory enzymes that control the citric acid cycle, we said at 3 sites, or 3 enzymes, or 3 reactions. First one is citrate synthase. Now, citrate synthase is an allosteric enzyme which is inhibited, as I said, by both ATP and NADH, also succinyl-CoA. Now, in addition to citrate, we said طبعا citrate here is a product inhibition, whereas succinyl-CoA here exerts feedback inhibition. Now the second regulatory site is the isosaturate dehydrogenase. Here, in this case, we see that the enzyme again is inhibited by ATP and ADPH, but however it is stimulated by ADP and NAD+, and these are allosteric activators of the enzyme. Now the third site here is catalyzed by the enzyme called Alpha-Ketoglutarate dehydrogenase complex. Again, ATP and NADH as well as succinyl-CoA inhibits the enzyme. Note that succinyl-CoA here acts as a product inhibition in this case. Now if we look at this table, which shows the relationship between the metabolic state of a cell and the ATP to ADP and NADH to NAD plus ratio in 2 states. First, cells in a resting metabolic state. Now in this case, there is no need to expend ATP, so ATP is high, ADP is low, and no need to use NADH, so it's high, and NAD plus is low. So the 2 ratios are high, and we call this energy charge. These ratios are called energy charge. So when energy charge is high, then this will shut down oxidative enzymes. And when cells in a highly active metabolic state consumes ATP, so low ATP, high ADP, and consumes NADH, low NADH and high this, so we have a low energy charge. This will turns on the oxidative enzymes. So as you can see that the relationship of energy requirements to enzyme activity is the basis for the overall regulatory mechanism exerted at a few key control points in metabolic pathways. Now we go to the so-called Glycosylate cycle. This is a related pathway, a pathway in plants and some bacteria that is an alternative to the citric acid cycle. We know that animals can convert carbohydrates to fat, but not fats to carbohydrates. Now, the glycosylate cycle bypasses the 2 oxidative decarboxylation steps of the citric acid cycle and routes isosaturate via glycosylate to malate. Now, the 2 key enzymes that are responsible for the ability of plant and bacteria to produce glucose from fatty acid are isosaturate lyase, which cleave isosaturate to glycosylate and succinate, and malate synthase, which catalyzes the reaction of glycosylate with acetyl-CoA to produce malate. Now, these 2 reactions in succession bypass the 2 oxidative decarboxylation steps of the citric acid cycle. So the net result is an alternative pathway called the glycosylate cycle. Now, In this case, if we look here, this is the first enzyme. Now, two molecules of acetyl-CoA enter the glycosylate cycle and give rise to one molecule of malate. So at the beginning, لاحظوا isocitrate by isocitrate lyase is cleaved to succinate plus glycosylate. Then glycosylate plus acetyl-CoA to produce malate. So two molecules of acetyl-CoA are consumed. Now the two carbon units, which is then converted to oxaloacetate. Now glucose can then be produced from oxaloacetate by the so-called gluconeogenesis. If we look here at this figure here, we see that this cycle or this pathway results in the net conversion of two acetyl-CoA. This is one which is used condensation between acetyl-CoA and oxaloacetate to form citrate, and then here isocitrate is cleaved, and then the malate synthase will synthesize malate. The second acetyl-CoA is consumed here. So we have two acetyl-CoA molecules are used to synthesize one oxaloacetate. Now, to synthesize one glucose, you need 4 acetyl-CoA. Now, where does this cycle occur? It takes place in glycosysomes. These are specialized organelles in plants. It is very important in germinating seeds. It occurs in glycosysomes and cytoplasm in yeast and algae, involved in the growth of plants in the dark. Now, the fatty acids stored in the seeds are broken down for energy during germination. Now, the fatty acids give rise to acetyl-CoA, which can enter into the citric acid cycle and go to release energy in the ways we have already seen. So, seeds are rich in lipids, which contain fatty acid. During germination, plants use the acetyl-CoA produced by the fatty acid oxidation to produce oxaloacetate and other. Once plants begin by photosynthesis and can fix CO2. Now, the citric and the glycosylate cycle, they can operate at the same time. In addition, acetyl-CoA can serve as the starting point of synthesis of glucose and any other compounds needed by the growing seedlings. Now, this cycle also occurs in bacteria, and this point is far from surprising. Why? Because many types of bacteria can live on a very limited carbon sources. They have metabolic pathways that can produce the biomolecules they need from quite simple molecules. So the glycosylate cycle is one example of how bacteria manage to do this. Now finally, what is the link to oxygen? Now the citric acid cycle is considered part of aerobic metabolism, but we have yet not encountered any reaction in this chapter in which oxygen takes place. So the reactions of the cycle are ultimately related to those of electron transport and oxidative phosphorylation, which do eventually lead to oxygen. Now the cycle acts as a vital link between the chemical energy of nutrients and the chemical energy of ATP. Now many molecules of ATP can be generated as a result of coupling to oxygen. And we shall see in the next chapter the number depends on the NADH and FADH2 generated in this citric acid cycle. So if you recall the classic equation for the aerobic oxidation of glucose, that glucose plus O2 is oxidized completely to water plus CO2. We have seen the metabolism of glucose through glycolysis. Now we see more of the CO2 comes from namely the 3 decarboxylation reactions associated with the citric acid cycle. Now in the next chapter we will see where the water and oxygen come from. End of part